welcome to my YouTube channel. So today we are going to discuss about, we are going to start module 4 of tissue engineering. So module 4 of tissue engineering speaks about basic biology of stem cells. Okay, so you might have heard about stem cells. For example, usually stem cells are nothing but cells which acts as a reserved cells which will be there in our bone marrow. Whenever these cells whenever our body loses cells, maybe because of damage or maybe something else, so our body produce these stem cells which is stored in the bone marrow. They will undergo differentiation. Okay, so stem cells are not at all active in state. They are like mother cells. They are there, but they are not specialized cells. They don't have any particular function to do unless and until if there is any requirement. Their only function is to undergo differentiation and give new cells. Okay, after differentiation, it will give you new cells. Those cells will be useful. For example, they are unspecialized cells, but when they go for a differentiation, they will give you specialized cells. Okay. So like that, if the stem cells, if before differentiation, okay, it is just a stem cells, like a pluripotent stem cells. But after differentiation, it can become a RBC, platelets, T cells, B cells, all these things. Okay, understanding? So that's what stem cell means. Let's see what this module has to say according to your syllabus. Stem cells definitions. The stem cells are unique cells they present in the body that have a potential to differentiate into various cell types or divide indefinitely to produce other stem cells. Understanding? So stem cells are nothing but unique types of cells that are present in our body that have a potential to differentiate into various cell types. That means, as I told you, it can be the blood cells, it can be the lymphoid cells like T cells, B cells, anything. Or that is about one function. Another function, they can divide indefinitely to produce other stem cells. Again, both the definitions are same. So stem cells are essential cells that replace damaged cells or cells lost due to the diseases. Maybe when the person gets disease, certain cells will be lost, maybe because of that, or maybe because of the damaged cells. Okay, so during these damaged cells, that has to be corrected, right? So the stem cells will undergo differentiation to give the healthy cells so that they can replace the damaged cells. So these cells are the earliest cells of the cell lineage in all the tissues and are found in both embryonic and adult organisms. So stem cells are nothing but earliest cells of all cell lineage. It means the first primary cells, what we can say. Okay, and they are found in both adult and embryonic organisms. So these cells provide a continuous supply of new cells that make up the tissues and as well as organs of animals and plants. That means these stem cells will give you some new cells. That's what we are discussing. After differentiation, it will give you some new cell types that make the tissue and as well as organ of the animals and plants. Okay, so stem cells have been of a great interest as therapeutic methods for various diseases and conditions, because wherever we need to replace the damaged cells, we make use of the stem cells. We can either extract it from the bone marrow and then you clone it in the laboratory, and depending on the disease, distinct characteristics and all, we can modify and then we can put it back into the human being so that these stem cells will undergo differentiation and go to give particular cells, maybe the blood or platelets, or maybe the other cells like lymphoid cells like T cells, B cells, islet cells, kidney cells, liver cells, any kind of cells, or thrombogenetic correlated thrombocytes. Okay, excuse me. For various diseases and conditions, whenever we need cells, the stem cells will provide cells. So stem cells in embryonic and adult organisms, if you can see in embryonic and adult organisms how they are, where they are, where we can find them, all those things we will see. So the stem cells in embryonic organisms are present in the inner cell mass of blastocyte. So there is something called inner cell mass, that is a blastocyte we call. Okay, so the embryonic cells, the stem cells in embryonic organisms we can find in blastocytes, that is inner cell mass of the blastocyte. So which then it differentiates into all other cells in the body. So the stem cells in adults, that was about embryonic organisms, whereas in adult organism, however, they are localized to specific areas within the body, like bone marrow, gonads and all. Okay. So pluripotent stem cells, here adult stem cells, adult blood stem cells and other stem cells. Adult stem cells on the pluripotent stem cell means it's unspecialized, undivided cells. When it undergoes time differentiation, it will give you the specific cells or specialized cells. So you can categorize into adult blood stem cells and other stem cells. Adult blood stem cells will give you a common lymphoid progenitor, okay, or a myeloid progenitor, whereas this common lymphoid progenitor will give you T cells, B cells, okay, and then for T cells we are having the NK cells, T cells, whereas B precursor we are having the B cells, okay. Under B cells again plasma cells, memory cells, all these things comes into picture in common lymphoid progenitor. Whereas in myeloid progenitor, specifically all the blood related cells, erythroblasts, which is nothing but RBC. Then we are having megakaryocytes for to develop the platelets, and directly to the other entities in the blood, like other blood cells, what you can call neutrophils, basophils, eosinophils, monocytes, all those things. So that was about adult blood stem cells. So speaking about other stem cells, we are having the muscle cells, okay. That is also being done by the other stem cells, and we can also develop the nerve cells, cardiac cells, liver cells, and also going for intestinal cells. So here simply what we are saying is, whatever this brown thing is there, no, purple, these all are mother cells, and other color cells are after differentiation they are getting different specific cells, specialized cells. The properties of stem cells. All the stem cells found throughout all living systems have three important properties. What are those? These three properties can be visualized in vitro by a process called clonogenic assays, where a single cell is assessed for its ability to differentiate. So that we will come to know that whether this particular cell is has got a potential to differentiate or not. Let's see those three properties. The first thing is capable. The stem cells has got capable of dividing and renewing themselves for a long period of time. So it's the first thing is capable of dividing and renewing themselves whenever there is the shortage. So these cells undergo a period of cells proliferation while preserving the undifferentiated state. So next one, we are having all stem cells are unspecialized or undifferentiated. So as I told you that these stem cells are unspecialized, undifferentiated. There is one more property. The first property, they have got a capability to divide, okay, and renew themselves. Second one is they are unspecialized, undifferentiated. When they undergo differentiation, they will give you the specialized cells. Second property. The third property is that ability to differentiate into specialized cells that together make up a different tissue types. Like these cells can be either pluripotent or multipotent. So these are the three different properties of stem cells. What is what? Capable of dividing and renewing themselves. Second one is unspecialized or undifferentiated cells. After differentiation, they will give you the specialized cells. Okay, so ability to differentiate into specialized cells and with different tissue types, and we can have two types, pluripotent and multipotent. This is exactly how the stem cell culture looks like. We are having a cleavage stage embryo. At this stage, we are having one cell here, the particular stem cell, the cultured blastocytes, inner layer of the cultured blastocytes, and we can isolate the inner mast cells which contains the stem cells. Now irradiated mouse fibroblast feeder cells, okay, so that cell dissociated and replaced. So once you add this particular feeder cells, cell will be dissociated. So new feeder cells can be added, so that again whatever the remaining cells are, that will be removed or dissociated. So that established embryonic stem cell culture. So we are having the only pure stem cells. Okay. So this was taken by the John Willing and Sons. Okay. And Haynes et al. Has done this particular work. From their paper, this image has been taken. Here I am giving credit to them. So stem cell culture. How exactly we can culture the stem cells? There is increasing interest in the optimizing stem cell culture, not only because such a culture is widely used in basic research for studying stem cell biology, but also owing to the potential therapeutic applications of captured or cultured stem cells. So because of their ability to replace damaged cells, what happens? These cells are cultured under artificial conditions to produce more of these particular cells, so that we can, whenever there is a damaged cells, we can replace. And we have got basically embryonic organism and adult organism. That's why we are having embryonic versus adult stem cells. Embryonic stem cells are more potent than the adult stem cells, as they are capable of differentiating into various cell types. Whereas adult stem cells, however, they are less potent compared to the embryonic, whereas embryonic more potent. Here it is less potent, that means less potential it has got to differentiate, and more restricted, as they can only differentiate into a specific lineages. Okay, only for specific lineages they will differentiate. Because of this reason, embryonic stem cells are mostly cultured and to obtain embryonic stem cells. So stem cell culture conditions must be defined according to the stem cell type. Example, cell culture requirements for embryonic stem cells, that is ES, or different kinds of adult stem cells may not be the same. So whatever the culture conditions is required for the embryonic stem cells or adult stem cells may not be the same, because they are different. Okay. Now the process of stem cell culture should be refined throughout the process, as stem cells always balance between self-renewal and differentiation. Always they differentiate, the balance between the self-renewal and differentiation. And the culture of the stem cells might be different from that of the other cells, as some stem cells require non-standard culture reagents such as feeder cell lines or cell layers, conditioned media, or growth matrices. So it depends on the several factors. It may be the feeder cells, okay, which we have seen in the images in the previous slides. Our conditioned media may be the media which we are using for the cells to grow, or growth matrices, okay, where these cells are growing. Okay, maybe scaffolds, what you can say. So different media and conditions are to be employed, obviously, considering the purpose of the culture, where some culture might be done to keep the cells in their stem state, while others might want the cells to be differentiated. So different media and conditions can be used, and also we can either go for all the stem cells to be differentiated, or only a few stem cells should be differentiated and other stem cells should be as it is, so that they should have that capability still. During the culture, the cells are usually passaged under the laminar flow hood, okay, and maintained in incubators which are under atmospheric partial oxygen pressure, okay, in order to maintain the hygiene, or in order to maintain if there should not be any inflammation, infection by any bacteria, microbes, or contaminations, we can use the laminar flow hood. So another challenge in the cell culture is that the control and as well as the maintenance of well-defined cell culture conditions. So imagine well-defined cell culture conditions are there. So to maintain that and to control that, it's very difficult and very challenging, especially pH and oxygen pressure. You cannot keep on controlling the same pH for a long period of time. It's very difficult to do that. Same thing goes with the oxygen pressure. So next we are having subtopic is types or sources of stem cells. We are having n number of stem cells. So majorly we are having embryonic stem cells, adult stem cells we just discussed, or you can say adult stem cells can be categorized into somatic or tissue specific stem cells. Third one is induced pluripotent stem cells. Fourth one is perinatal stem cells, and fifth one is mesenchymal stem cells. Importance of stem cell research. The process of cell differentiation, stem cell based therapies, stem cells to renew or test the new drugs. Applications. Stem cell research has been used in various areas because of their properties. Okay, self-renewable and ability to differentiate into to give rise into a different cell lineage, different cell types, all those things. So some of the common applications are here. So first one is stem cell research has been used in the field of regenerative medicine. What do you mean by regenerative medicine? Regenerative medicine is something you are regenerating with the help of already existing things. For example, stem cells are already existing. Okay, they are naturally existing, and you are using it for again in order to build or treat damaged cells. Okay, that's why it is called as regenerative medicine. The field of regenerative medicine, which deals with the restoration of the tissues or organs in the patient suffering from the severe injuries or some chronic diseases. The next application is the progress made in the field of stem cell research has laid the foundation of the other cell-based therapies of disease that cannot be cured with the conventional medicines. Okay, you can go with this particular whatever the conventional method can be used, and if you are not able to come up with the solutions, you can also go for the stem cell therapy also. So studies related to human stem cells research has enormous potential for contributing to our understanding of the fundamental human biology. So the fourth application is many years of research on stem cells have made it possible to transplant hematopoietic stem cells to the patients after cancer treatments. So that if any blood cells are, because many blood cells has to be very active, and it should be more pure and hygienic. So we can have the more proper healthy blood cells after cancer treatments. And stem cells research has also been used for the testing of new drugs, okay, before they can be tested on animals or human beings. So what happens before we test on animals or human being, we can always try on in particular laboratories or in vitro, or by using some computational techniques, like bioinformatics tools and techniques. So that can be done with the help of stem cell research. And also, in the laboratory, if a particular person has got some particular disease, I take that particular cells and keep it in laboratory, and I try to understand what kind of cell I want, and then with the help of stem cells, I will get that cell, and then I will introduce that cell back into the human being. Because sometimes not all the patients will be having capability of differentiating stem cells, differentiation of the stem cells to give rise to the required stem cells. Sixth application, cultured stem cells are used for the transplant of the cells in the case of various diseases like bone marrow for leukemia, nerve cells for Parkinson's disease and Alzheimer's diseases, heart muscles for heart disease, pancreatic islets for diabetes. These are all the different cells I can get with the help of stem cells in order to stop all these diseases. These are the cells we need, these are the diseases we will come to know if any fault or if any missing function or if any malfunction in these particular cells. Limitations or challenges of stem cell research. The most crucial challenge to the stem cell research is that ethical related issues. Okay, especially when it comes to the embryonic stem cells, because the ethical guidelines has to be followed by everyone. Okay, so that is very important. So the source of the some stem cell lines might have mutations which increase the chances of the mutation in the transplant. The source of the some stem cell lines, usually the adult stem cells, they might have a mutation, particularly what is mutation, so which increases the chance of the mutations in the transplant. For example, imagine the stem cells I am bringing from a bone marrow from some person, and I am using it for another person. Imagine that stem cell has got mutation already, then imagine the condition, because if the mutation is there in stem cells, then when it undergoes differentiation, it will spread that mutation. on entire genome or cellular level. So that is going to be again a problem. Okay. So it is also difficult to transplant the stem cells produced in the laboratories to the target cells. It's very difficult. We cannot just simply copy paste or something like that. It takes time. It takes time. It takes effort. All those things. So that is also one of the challenge. Embryonic stem cells also do not permanently renew themselves in vivo. That is also one of the problem. We cannot say completely all the stem cells will renew themselves, but embryonic stem cells do not permanently renew. Okay, at some stage they will stop renewing themselves. That is also one of the challenge. Next challenge we are having self-renewal of these cells can be achieved in vitro under the artificial conditions which inhibit their differentiation. Sometimes artificial conditions may inhibit the stop the differentiation of the stem cells. It is also challenging to obtain a sufficient amount of the stem cells with the ability to differentiate into a desired cell types. So it's obviously in order to get sufficient amount of cell that again is very difficult to get. The next challenges we are having involves a certain amount of the spontaneous differentiation into the various cell types which might not require but that spontaneously the differentiation occurs. So we cannot have a control over it. So additionally there is one more challenge. Additionally there is one more challenge, the last challenge. The differentiation is not synchronizable yet. We cannot manipulate it. We cannot tune it. Okay, leading to a mixture of the cells in various stages of the development. So next topic we are having hematopoietic differentiation pathway, or we can call it as hemopoiesis. We will study in the next class.